In 1986, a neuroscientist at the University of Southern California had an idea the scientific community thought was somewhere between unlikely and insane. He wanted to put serial killers in a brain scanner, not to rehabilitate them, not to treat them, to photograph their brains while they were alive. It took years of bureaucratic battles, ethical reviews, and institutional resistance to make it happen. When those first scans came back, what Adrian Raine saw changed the trajectory of forensic neuroscience permanently. The brains were different, not slightly different, not ambiguous, starkly, measurably, unambiguously different, in the very regions most critical for empathy, impulse control, and guilt. Today we go through what that science actually shows, region by region, study by study, evidence by evidence. This is not armchair psychology. This is not sensationalism. This is peer-reviewed neuroscience. And it is more disturbing and more important than almost anything the true crime genre has ever covered. If you haven't watched episode 1 yet, go do that first. It sets the foundation for everything we're building today. For those who are back, welcome to the deep end. Last episode we asked the question. Today we start proving the answer. Let's start with how we actually see inside a living brain, because the tools matter, and understanding them makes what we found 10 times more powerful. PET scanning, positron emission tomography, works by injecting a small amount of radioactive glucose into the bloodstream. Since the brain runs on glucose, active regions consume the most. The scanner maps it in real time. Active regions glow hot, reds and oranges. Less active regions show cool, blues and greens. Functional MRI measures blood flow and oxygen in real time. Active neurons demand more oxygen, higher spatial resolution than PET, no radioactive tracers. It became the dominant tool from the late 1990s onward and revealed detail the earlier scans couldn't reach. And then there's diffusion tensor imaging, DTI, perhaps the most fascinating tool for our purposes. It maps the white matter tracts, the actual physical cables connecting different brain regions. It lets researchers see not just whether regions are intact, but whether the connections between them are healthy, degraded, or cut. 4 tools: PET, fMRI, structural MRI, DTI. Together, they give us a window into the living brain that no generation before us has had. And what 30 years of using them on convicted killers has produced is a pattern, consistent, reproducible, confirmed across dozens of independent research groups worldwide. The pattern is real, and it is specific. Let's go through it, region by region, starting at the very front of the brain and working our way deeper into the darkness. The prefrontal cortex, forward third of the brain, just behind your forehead. In evolutionary terms, it's what makes us most distinctly human, the region most dramatically expanded in Homo sapiens compared to every primate we're descended from. Planning, abstract thinking, impulse inhibition, moral reasoning, the ability to mentally simulate the consequences of your actions before you take them. It's your brain's boardroom, where the future gets considered before the present acts. Adrian Raine's landmark 1997 study, published in the Archives of General Psychiatry, compared the brain scans of 41 murderers against matched healthy controls. The finding? Significantly lower glucose metabolism in the prefrontal regions of the murderer group. The boardroom wasn't just underperforming. In many cases, the lights were barely on. A 2019 study in Neuroimage pushed even further, finding reduced gray matter volume in the ventromedial prefrontal cortex of persistent violent offenders. Not just less active, physically smaller, less tissue, less infrastructure. The boardroom isn't just dark, it was never fully built. What does reduced prefrontal function actually look like in behavior? It looks like impulsivity without consequence evaluation. Acting without the mental simulation most of us run automatically, the process that asks, if I do this, what happens next? Is there a cost I'm not willing to pay? For most people, that simulation runs constantly and unconsciously. It's why you don't say the cruel thing you thought of, why you don't take what isn't yours, why you stop. For people with severe prefrontal dysfunction, the boardroom is dark, and the decision gets made in the hallway. Nested within the prefrontal cortex is a subregion that researchers now regard as perhaps the single most critical area in the neurobiology of extreme violence. It sits directly behind your orbital sockets, your eye sockets, the orbitofrontal cortex, the OFC. The OFC is the primary neural site for behavioral inhibition, the ability to stop yourself from doing something even when part of your brain wants to do it. This is where social rules are enforced at a neurological level, where the learned consequences of past behavior are automatically applied before new impulses fire. Patients who develop OFC lesions through stroke or brain injury, people with completely normal personalities beforehand, frequently undergo dramatic personality changes. They become crude, impulsive, indifferent to consequences, unable to self-regulate. Not because they chose to change, because the brake was removed. Neuroscientist Jim Fallon at UC Irvine spent decades scanning killer brains. His findings stopped the field. OFC dysfunction appeared in every single case he examined. Without exception, without one outlier in the entire group. He began describing it as a near-necessary component in the neurobiology of extreme violence. And then came the moment that changed Fallon personally. Years into his research, he agreed almost casually to have his own brain scanned as part of a family Alzheimer's study. When the results came back, his own brain scan showed OFC activity patterns that looked remarkably similar to the killer profiles he had been studying for years. Fallon's conclusion from his own experience is perhaps the most important line in this entire series. The brain loads the gun, but the circumstances of your life, your relationships, your structure, your choices, determine whether it fires. Neurological risk is not neurological destiny. The orbitofrontal cortex, the brake, present in every healthy brain. Compromised, measurably, consistently, reproducibly, in the brains of people who commit the most extreme violence. That is not an opinion. That is a replicated scientific finding. Deep in the medial temporal lobe, buried beneath the cortex, sit two almond-shaped clusters of neural tissue, the amygdala, ancient. In evolutionary terms, one of the oldest structures in the mammalian brain, predating the cortex by hundreds of millions of years. The amygdala is the primary biological substrate for empathy and guilt. When you wince watching someone stub their toe, when you feel dread seeing someone in danger, when you lie and feel that sickening twist, your amygdala is firing, generating an aversive emotional signal in response to harm. Studies of psychopathic individuals, a population with substantial overlap with serial offenders, have found a 5 to 18% reduction in amygdala volume compared to healthy controls. The structure is physically smaller, less volume, less capacity, less guilt. But volume alone doesn't tell the whole story. Functional imaging shows that even when the amygdala is normal in size, it often shows dramatically abnormal responsiveness in psychopathic individuals. Show a normal subject images of suffering, their amygdala fires intensely. Show the same images to a psychopathic subject, flat, muted, nothing. The clinical result is something both documented and philosophically staggering: a reduced biological capacity for guilt, fear, and empathic distress. The feedback loop that makes cruelty aversive, the loop that stops the overwhelming majority of humans from extreme violence even when angry, even when they have opportunity, is muted, inconsistent, or absent. Without that loop firing, harm becomes consequence neutral at a neurological level, not as a choice, not as a philosophy, as a biological fact. The thing that makes cruelty painful to the person committing it simply isn't there. The amygdala, two almond-shaped structures, physically smaller, functionally muted. The guilt generator that doesn't generate. The empathy circuit that doesn't complete. And we haven't even gotten to the worst part yet. Now we get to perhaps the most important finding of all, and it's the one that most true crime content never reaches. Because it's not just about individual brain regions being damaged, it's about connection. The circuit that allows your emotional brain to talk to your rational brain. The uncinate fasciculus, the primary white matter tract connecting the amygdala to the orbitofrontal cortex. Diffusion tensor imaging studies consistently find reduced structural integrity in this specific tract in serial killers and psychopathic individuals. The cable is degraded. The signal is weak. The two regions that should be in constant dialogue are functionally isolated from each other. Think about what that means with one simple metaphor. Your smoke detector works. Your sprinkler system works, but the wire connecting them has been cut. The smoke detector detects fire. The sprinkler can deploy water. But without the connection, one will never trigger the other. The fire keeps burning. In a normally functioning brain, the amygdala detects harm to yourself, to another, and sends a distress signal to the prefrontal cortex, which generates guilt, activates behavioral inhibition, and puts the brakes on the action. The circuit closes. The moral override fires. In the killer brain, the signal is generated, but it never reaches the brake. The action continues. The cable is cut. 3 words that summarize the central neurological finding of 30 years of forensic neuroscience research into extreme violence. The cable is cut. There is one final brain region worth understanding, rarely mentioned in true crime content despite being highly relevant. The insula, buried deep in the lateral fold running horizontally along the side of each brain hemisphere. The primary neural site for interoception, your brain's real-time awareness of your body's internal state. The insula is central to the experience of disgust, physical revulsion, and social empathy. When you feel viscerally sick at the sight of something deeply wrong, when your body physically recoils from witnessed cruelty, that is your insula firing. A powerful secondary brake that reinforces what your prefrontal cortex tells you rationally. This is wrong. Stop. Multiple peer-reviewed studies of violent homicide offenders have found reduced gray matter density in the anterior insula. Less tissue, less function, a muted or absent disgust response. The body no longer provides the visceral reinforcing signal that would function as an additional and powerful barrier against committing extreme violence. Let's put the whole picture together. Reduced prefrontal gray matter, the consequence simulation center. Damaged orbitofrontal cortex, the behavioral brake. Reduced amygdala volume and function, the guilt and empathy generator. Degraded uncinate fasciculus, the connecting cable. Reduced insular cortex, the disgust signal. All 5, in the same brain, at the same time. Now, the most important thing I'll say in this entire episode. This science does not prove that anyone with these neurological features will become a serial killer. Millions of people have reduced prefrontal gray matter. Millions have smaller amygdalae. and the overwhelming majority live completely ordinary, peaceful, productive lives. Neurological abnormality is not neurological destiny. What the science proves is a measurable biological substrate for the behavioral profile of extreme violence. What it suggests, with enormous implications, is that identifying these risk factors early, combined with developmental support, might one day offer a path toward prevention our current systems simply cannot access. The brain loads the gun. Life decides whether it fires. That is the core finding of 30 years of forensic neuroscience, and it is as hopeful as it is disturbing. In episode 3, we leave the laboratory and enter the crime scene. Every neurological finding from today gets mapped directly onto the documented behavior and confessions of 4 of the most significant killers in history. Before episode 3, I need your answer in the comments right now. Which finding from today's episode hit you the hardest? The Fallon personal brain scan? The cut cable? The 18% amygdala reduction? Drop it below. I read every single one. If this episode changed how you think about the neuroscience of violence, hit like right now. It takes 2 seconds, and it is the single most powerful thing you can do to get peer-reviewed science in front of people who need it. Subscribe and ring the bell. Episode 3 drops next week, and you do not want to miss it out of sequence. Episode 3 is up, or dropping very soon. Click it now. The brain science you learned today is about to walk into 4 real crime scenes, and it will change how you see every one of them permanently.